Hi, welcome to Protein Synthesis Land. We'll be your tour guides. I'm Danielle.
I'm Emily.
Jacob.
Okay, so to start, we're going to start with transcription.
Transcription is how RNA is made, which is made from the DNA. And the polymerase starts off by breaking the DNA one by one. And it'll break all of the hydrogen bonds. And then the RNA strand, the RNA is made. What? The RNA is made by one, and it starts at the promoter. And it starts at the promoter, which is, according to Buffalo State faculty, is a DNA sequence that controls the efficiency site and direction of the action. And it breaks apart, and then they come together one by one and match with the complementary bases, the complementary strands. And the uracil is used in place of thymine. And as soon as they come together, it stops at the stop.
It just stops. Alright, so after the RNA strand is made, then the proteins are made, because that's the whole reason that it happens. So what we have, the little trains, are because a good way to think about it is that the RNA is like a train. It moves along, passengers come, passengers go. It has stops, which would be like the ribosome, and the nucleus would be like the train station. So the RNA strand comes in, and so then when it's in the ribosome, the RNA strand comes in. So the first anticodon, to match with the start codon, would come in carrying methionine, and it would go through the ribosome. And then the second RNA would come in. The second tRNA would come in, and it would carry that. And then the amino acid would break off and get attached to the peptide bonds, and this RNA would leave. And these would get attached, and this tRNA would stay. And that process would continue for all of them, so as it moves down. And then this tRNA leaves, and it just goes back to carry out a new function later. And that would continue for the rest of the molecules.
And then once the ribosome reaches the stop codon, so the stop codon we have here is it's UAG, and this has no anticodons or tRNA, and so it's over here, and then we have a polypeptide bond, which is made up of the amino acids, and this falls off, so we have a very long chain of proteins, and the tRNA will leave the ribosome, as it already did, and then the ribosome just moves away from the mRNA, and the polypeptide bond is released, and you have a protein.